In this next video, I would like to walk you through actual activation of a T cell and actual activation of a B cell, because there are steps. You know the general outline of this, but I want to show you the specifics and write out the specifics and tell you where in the body it happens. So let's get to it. We'll start with T cell activation. All right, so the first thing we start with is an immature T lymphocyte. And at this point, we don't, it hasn't chosen a co-receptor. So it hasn't committed to being CD4 or CD8. So we don't say what kind it is. But let's pick one. Let's choose one. So let's say this is going to be a cytotoxic T cell, CD8 positive cell. That's one of my favorites. So it goes through education. T cells are educated in the thymus, and specifically of children and adolescents. So that's where education happens. If you're an adult or adult-ish, chances are you're done educating your T cells. So remember I said that a lot of cells die during this process. It is a brutal process where a lot of cells get recycled and become cell material because they don't pass through this process. But we'll talk about that in another video. Here I just want to say our little cell passes through education and it becomes a mature, naive, CD8 positive T cell. Okay, so that's what it is after it succeeds in education. And what happens in that education, we'll talk about in a little bit. So then that CD8 positive cytotoxic T cell, what happens to it? It waits. It waits. This is what you should picture. I don't know if you can see this or not, but you should go to histologyguide.com and find the lymphoid organs section and just zoom in on that lymph node, because it's amazing. Every little dot in here is a lymphocyte, and towards the middle of the lymph node, these areas with all this tissue, to my understanding, this is where dendritic cells show antigens to mature naive T cells. This is where dendritic cells try to activate T cells. And every little dot here is a lymphocyte. And there are so many in every lymph node. And they're all hoping that they will get activated. They're all hoping they will recognize an antigen. So, a dendritic cell, which I'm abbreviating DC, the dendritic cell shows it the correct epitope. This is super rare. Most dendritic cells have shown it the wrong thing, but at some point, maybe, a dendritic cell will show it the thing it recognizes. This is typically only going to happen during a real immune response, when there's a ton of some pathogen in the body. That makes this likely. Only if a dendritic cell shows it its epitope, then it will go through activation. You know what? I'm an idiot. I shouldn't have called this activation. I should have called this development. My apologies. So activation, this is what happens now, and you can draw the next two arrows because one of them is going to be clonal expansion, which is where we go from one of these T cells to many of those with the same receptor, and then they differentiate. You go through differentiation. And that's when you get two different types of cells. One is going to be a memory CD8 positive T cell, which is kind of going to act as if it were a mature but naive CD8 positive T cell, because they wait to be activated. And that's what a memory T cell is going to do. It's going to wait for the next time the body sees this pathogen, a year later or 10 years later. And then there will be effector cells, and these are going to be the CD8 positive cytotoxic T cells. And these are the ones that kill virion factories for us. So, this is how a T cell develops. If it's going to go all the way through development, big questions here. Will it successfully get through education, and then will it ever activate? Only 5% of them successfully get through education, and a vanishingly small percentage ever activate. Most of them never activate. So if you were going to do this for a CD4 positive T cell, the only difference would be it would make a CD4 positive memory T cell, and then depending on what the body wants at that time, it's either going to make type 2 or type 1 helper T cell. That's not determined until this process begins. And we haven't seen what determines that yet. So that is T cell development. Let's look at B cell development. Wow. I should never write things on this graphics pad. But what are you going to do? You work with the tools you have, you know what I mean? So B cell development. You're going to see this is different from T cell development in one important way, and I also want to make a very important connection for you. So let's get into it. You start with an immature B lymphocyte, and it's going to go through education where? In the bone, in the red bone marrow. And if it survives, it will become a mature, naive, mature but naive B cell. So now B cells are very different from T cells because a B cell receptor doesn't need to participate in antigen presentation. So I draw some B cell receptors. They are, if you looked at them like their actual shape, they would just look like antibodies that are embedded in the membrane. So this is very much a conceptual cartoon drawing. But if you're showing a B cell finding its epitope for the first time, it's not part of antigen presentation. It just bumps into it. Boom. Found it. And so that is like minor, we'll call that minor activation. And this is when the B cell bumps, literally bumps, that's a P, into its epitope. This could happen in the blood, in the lymph, wherever that B cell is. If that happens, you get minor activation, and the B cell makes some antibodies. That's all it can do. It can't go through clonal expansion or anything like that. And so now I'm going to call this a mature, naive-ish B cell. It's not really naive because it has seen its epitope. And naive lymphocytes are the ones that have never seen their epitopes. So it's not really naive, but it's also not activated. What it needs to do is perform phagocytosis on this whole thing and process it and then display parts of it on MHC2 to a certain type of helper T cell, which you should be able to tell me. That's what it needs to do. So if it's going to present its antigen, whatever it attached to, on MHC2 to a helper T cell type 2. And if the helper T cell type 2 recognizes it, then it will activate. It'll go through full activation. It'll go through clonal expansion. It'll go through, or its daughters will go through differentiation. And so what do you get when a B cell goes through differentiation? You get memory B cells that give you your immune memory, and you get plasma cells. I don't know where the name plasma cell comes from, but it doesn't tell you anything useful. What plasma cells are, you know they make antibodies, but like, they make antibodies. They are designed for one purpose, and that is to make huge quantities of one antibody. So once you have plasma cells in an immune response, the body is flooded with one antibody, endless copies of one antibody. And that can be very powerful part of the immune response. So this is lymphocyte development, and I just wanted you to see it a little more specifically because this is an opportunity to connect to other things in the immune system. So the last thing I want to talk about is education. So lymphocyte education. And really there are just two processes here. I'm going to type this because I have to type this. Specialized epithelial cells show the immature lymphocyte every protein they can make. Period. We try to show them every possible human protein. Okay, that's what happens in education, and what determines whether they survive is how they respond. So the first question they get asked is, do they interact correctly with the antigen? Do they interact correctly with it? And I don't know the details of this. What I've read is that it's like, do they exchange the right signals to show that they have looked at it? Like, are they able to actually look at an antigen? Does their receptor work as a receptor? So it's basically checking them out to see if they have a functional lymphocyte receptor system. And so if they do, yes, 10% of them roughly have that whole functional system and they survive. 90%? No. They die and are recycled immediately into new cells. So their cell parts get used immediately by the body. So if they survive, then they have a second test they go through. And this is, did they recognize any of the antigens? Okay, so first, do they interact correctly? Can they look at an antigen? And if yes, good. Then we ask them, do you recognize any of the ones that a random epithelial cell can show you? And if yes, this is about 50%, they die. If no, which is about 50%, then they become mature, naive lymphocytes. And that's lymphocyte education. It's really two questions they get asked. Do you correctly work as a lymphocyte? Yes, you survive. 90% of immature lymphocytes have some problem and get killed. And then the ones that are successful lymphocytes, do they recognize? Does their receptor recognize any human proteins? If yes, wow, they're going to cause an autoimmune disease because they're going to eventually be exposed to that human protein, and they're going to act however they act when they're going to do an immune response, whatever they do. We don't want that. We don't want antibodies against actin. We don't want antibodies against hemoglobin. We want none of that. So we kill the cells that can recognize human antigens. And what we're left with is a pool of mature naive lymphocytes, billions of them in lymph nodes all over the body, that we have no idea what they recognize. They were randomly generated. We just don't know anything about them, but we know they are lymphocytes. They are ready. They are waiting to activate. So that's it's a cool process because it just makes sure all the lymphocytes we have work and none of them are going to kill us. So thank you for your attention to this. I'll see you at the next video.